First of all, I'd like to thank the organizers of the symposium for including me in this format. And what I'd like to talk to you about today is some of our work on hepatitis B virus. So as you can see, the title of my talk is A Mouse Model of Human Hepatitis B, a Tool to Identify and Test Therapeutic Targets. So this may be familiar to many people in the audience, but I just still want to start by making a pitch that despite the fact that there's an effective vaccine, hepatitis B is still a major global pathogen. It's a small double-stranded DNA virus that causes acute and chronic hepatitis. 1/3 of the world's total population approximately has been infected with it. There are approximately 400 million people who are chronically infected with it lifelong. There's 1 million deaths annually from hepatitis B-related liver disease. 80% of hepatocellular carcinoma is attributed to hepatitis B. And in the United States, there are 1.25 million people chronically infected. So it's also a disease of this country, and particularly in the Bay Area, where we have a lot of immigrants from Southeast Asia. So hepatitis B is an immunologic disease. The virus infection itself isn't cytopathic, and it's the immune response to the virus that either controls viral infection and leads to clearance, or it's also the immune response that leads to the necroinflammatory process that's involved in chronic hepatitis, cirrhosis, and hepatocellular carcinoma. So the scientific and clinical goals of the hepatitis B part of my laboratory are to identify the immunologic mechanisms involved in pathogenesis of chronic hepatitis B infection, and to understand how these mechanisms differ from those generated during successful viral clearance. And the idea is to identify new therapeutic targets that can tilt the immune response in chronic infection towards clearance, and to actually work with the collaborators to develop new therapeutics. So our approach has been to develop robust experimental models to study HBV clearance, persistence, and disease pathogenesis, and to use the models to develop focused hypotheses to be tested in humans, and then to develop the clinical and human immunologic research infrastructure that really enables us to be able to study patients. So enrolling patients with acute and chronic hepatitis B, clinical database management, blood banking, nucleic acids, and liver tissue. And I'd say we've made huge progress in terms of a liver biorepository in the last 5 years in collaboration with California Pacific Medical Center. So the first thing that I want to talk about is developing the mouse models of primary HBV infection, which I've been working on for more than 15 years. And the first thing to know about hepatitis B is it doesn't infect mice. And mice are really the animals where we can study immunology and immunopathogenesis. And there are ways that people are trying to get around that to be able to infect mice. But what we've generated is a transgenic mouse model of hepatitis B that mimics certain important aspects of primary infection. So what we do is we use HBV, 2 lines of transgenic mice. One that has the HBV envelope protein, small, middle, and large, driven off of the albumin promoter. Only large envelope comes off of the albumin promoter, but the antigen is produced in the liver and then, of course, secreted. And then the other line of transgenic animals are the HBV replication mice. They have an overlength construct of the full HBV genome, and by virtue of this transgene, they actually are able to make infectious virus particles that are secreted. And by infectious, I mean infectious to, we know chimpanzees, we've never tested it in humans, but the mice, of course, aren't reinfected because there are a lot of blocks to reinfection and replication in mice. So these mice are perfectly fine, lifelong, again, demonstrating that the virus itself isn't cytopathic. And they don't mount an immune response to the virus because, of course, they've seen it through all of development, and they see the virus and viral antigens as self. And so in order to get rid of the tolerant adaptive immune system in these animals that don't want to mount an immune response to HBV, we cross them to immunodeficient mice, RAG mice. So these mice have no B cells, no NKT cells, and no T cells. And so what we end up with is a mouse where antigen, either the envelope protein or virus, originates in the liver in the absence of an adaptive tolerant adaptive immune system. And then to mimic the point of primary infection, we put in an entire spleen. So syngeneic naive splenocytes, a whole spleen, to put in a naive immune system into a mouse full of virus, and we monitor the immune response and pathologic outcome. And I'll say that this reconstitutes all compartments of the immune system and lifelong. So we spent a lot of time in the first few years in the startup of my laboratory looking at the activation of natural killer and natural killer T cells in response to HBV. And those studies were very interesting, and I still believe that these cells are important in disease pathogenesis. But I was really interested in studying cells and molecules that were crucial in determining clearance and persistence. And it turns out, although those cells may help tilt the immune system one way, they weren't pivotal. In order to develop a model that we really thought mimicked human HBV clearance and persistence, we used the fact that the main known predisposing factor influencing HBV disease outcome is the age of infection. So most infected adults mount an immune response that leads to viral clearance without any long-term sequelae. So maybe 5%, and maybe not even that many, of infected adults will get chronic infection. So if you weren't vaccinated and if you were exposed to HBV, you would mount an immune response likely that you'd have no problems. But 90% of vertically exposed neonates, so at the time of birth and shortly after birth, will develop chronic infection. And 30% of exposed children ages 1 to 5 will develop chronic infection. This is really different than any other infectious disease epidemiology that we know. There's something different about hepatitis B that allows it to become persistent and in young. And it's this group that harbors the major global reservoir of HBV because they harbor it chronically and then disease becomes clinically evident maybe 30, 40 years later. So based on some data that we had that there was age-dependent expression of certain genes by microarray, we decided to test what is the effective age on HBV disease outcome in our model. And so there are obviously some limitations with using this transgenic model, although we try to mimic key aspects. But one of the real benefits of this model is we can change the age of the immune system separately from the age of the recipient animal. And so what we did was to take different ages of HBV transgenics, and this is both the envelope and replication mice. And we used either our normal adult mice that we know generate an effective immune response to HBV, or we used young pre-weaned mice 3 to 4 weeks of age. And the reason we chose 3 to 4 weeks of age is 2 reasons. One is that I have to do tail vein injections, and that's about the age I can get it. But the other real reason is a lot of the dogma out there explaining this age-dependent difference in HBV disease outcome had to do with neonatal tolerance, that the antigens crossed the placenta and that there was this window where when the antigen was seen that you wouldn't mount an immune response to it. So 3 to 4 weeks of age is clearly out of the window of neonatal tolerance. It's the equivalent to a 6-month to 1-year-old human. And it's been shown in mice that it's out of the window of neonatal tolerance. And so in our systems, if we see a difference, it's likely not attributable to neonatal tolerance. So into these mice, we gave an adult immune system. So we're just comparing the hepatic microenvironment or the whole animal microenvironment that's different by age. So when we transferred adult cells into adult HBV envelope mice or replication mice, we got the hepatitis that we always see. So this is ALT, which is a measurement of hepatic necrosis. But we did not see hepatitis in the young mice that received the adult cells. It wasn't that the young mice didn't develop some inflammation, because in the adult mice, what you see is there is inflammation and necrotic hepatocytes. But the young mice still have inflammation greater than the non-transgenic RAG litter mates that also receives an adoptive transfer. So you can see the adults have the most inflammation, the young mice have less, and the RAGs less. And there is in these mice HBV-dependent cytokine production in both young and adult. But if you look at the other key markers of HBV clearance, one of them being clearance of surface antigen, what you can see is that in the envelope mice, the majority of the adult mice clear HBV surface antigen lifelong. The young mice never do. And in the replication mice, same thing. The adult mice clear surface antigen. The young mice don't. Other markers of HBV clearance in humans are the production of the antibody to surface. In fact, that's the target and the antibody produced with the vaccine. So what you can see is that the adult mice produce the antibody to surface, the young mice don't. But the real experimental result that convinced us we were onto something important is both the young and adult mice made an antibody response to core. And that's exactly what happens in humans. Young infants and children who become chronic still make an antibody response to core, but don't make an antibody response to surface. So what we ended up with is basically the serologic profiles of human clearance and persistence. The adult mice that got adult cells develop hepatitis, clear surface antigen, make the antibody to core, and make the antibody to surface. The young mice don't get hepatitis, don't clear surface antigen, don't make antibody to surface, but actually do make the antibody to core just like infants. So one of the other signatures and differences of HBV clearance and persistence is the strength and diversity of the T cell epitopes of the virus that are recognized. It's known that in people that are persistently infected, they do have an immune response and a T cell response to HBV, but the epitopes that are recognized are narrow. And in a response that leads to clearance, the number of epitopes and the strength of the response is much greater. So we tested this in our adult and young mice by using a peptide library that covers the surface antigen. So we had 15 MERS, 95 peptides, and 7 pools. And what we did was we take the lymphocytes from the livers of the mice at different time points, and we put it with the different peptide pools or no peptide pools, along with antigen-presenting cells, and then we count the number of spots that are making interferon gamma in response. And what we found was that both the young and adult mice mount a T cell response to the known dominant epitopes, but the diversity of the epitopes recognized in the adult mice is greater than the young, just like the difference between clearance and persistence in humans. This response was even more pronounced 3 months later, where again, major epitopes are recognized, but diversity in the adult and the mice that cleared is greater. And at 1 year, we could only find responses in the adult mice. So our conclusions from this is that we've developed an age-dependent mouse model that phenocopies key aspects of human HBV viral clearance and persistence. And this model can be used to identify key mechanisms that drive clearance or persistence and help us develop new hypotheses to be tested in humans. So now I'm just going to summarize a lot of data because we've been studying this for years to tell you some of the punchlines of the work. And the first is that, and this is published along with the model itself, is that T follicular helper cell production of IL-21 in the liver is crucial for an effective immune response to HBV. So without T follicular helper cells or IL-21, an adult mouse will mount an immune response that very closely mimics viral persistence. And T follicular helper cell number and IL-21 production is greatly diminished in the young mice. So T follicular helper cells make IL-21, which leads to a broad, multi-specific, and long-lived T cell response, and expansion and differentiation of B cells, which leads to the serologic signature of clearance, which is HBs antibody positive, core positive, and clearance of antigen. But in the absence of IL-21, we don't make antibodies to surface. We do make antibodies to core because we know that both adults and young do this, but we remain antigen positive. So we also went on, because we are interested in studying human disease and we're modeling a human disease, to try to validate our mouse model by testing focused hypotheses in humans. And what I can tell you is that we compared IL-21 expression in the PBMCs of patients with acute hepatitis B compared to those with chronic hepatitis B, but that are in the midst of a hepatitic flare. So their ALT levels and their viral loads are similar. So an immune response is going on in the liver. We also looked at carriers, so they have the virus, but there isn't inflammation in the liver, and uninfected. And what you can see is that there's a significant increase in the expression of IL-21 in adults who are acutely infected who go on to clear the virus. So the implications of our model from these results are that immune priming to HBV is occurring in the liver, and that effective immune priming to HBV is impaired in young mice. So we went on to say, okay, is immune priming occurring in the liver, and can we detect it and see it? And so we did our normal adoptive transfer model, young mice or adult mice with adult cells, and we assayed by ELISpot for HBV-specific responses days 1, 2, 3, 5, and 7 after adoptive transfer in the liver, the spleen, and lymph nodes. And what we found very clearly was that by day 3, we could detect an immune response in the liver, but we didn't find an immune response in the hepatic lymph node, which drains the liver. We didn't find it in the spleen, and we didn't find it in the mesenteric lymph node, which is pre-circulation to the liver. By day 8, again, we saw expansion of the HBV-specific T-cell response in the liver. Again, nothing in the spleen and lymph nodes. But by day 12, a lot in the liver, and it's only by day 12 we begin to detect it in the hepatic nodes, the spleen, and the mesenteric lymph node, saying that the immune response is initially detected within the liver. In addition, the HBV-dependent IL-21 expression is liver-specific. So this suggested to us that immune priming was occurring in the liver, so we went looking for it. And what we found, and I don't know if we can dim the lights so that you can see this, because I really can't see it on this screen. If you look at day 0, F480 marks the macrophages. The young and adult mice have similar number of macrophages and look very similar, if you could see it. I don't know if you can. But by day 8, at the peak of the immune response, you see these big clusters of macrophages. And co-localized to those clusters are CD4 cells, CD8 cells, and B cells. In the young mice, you still see F480 positive cells. You still see CD4 cells, B cells, and T cells, although reduced in number. But there isn't this lymphoid organization and clustering and co-localization of the immune cells. This is just a quantitation of that. You can see very significant difference in the number of macrophage clusters, very significant difference in the numbers of CD4s, CD8s, and B cells that co-localize, and very significant difference in actually the clusters of CD4 and CD8 cells, suggesting that they're expanding within the liver. So the other thing is that within the adult and young liver, there's diminished numbers of class switch B cells. So we see plenty of we see equivalent numbers of IgM positive cells, which are in green. But we never see any of the IgG positive cells in the young liver. And you can see this big difference in IgG positive cells. Again, suggesting that class switching and differentiation of B cells is occurring within the liver. So the conclusions from this is that immune priming to HBV occurs in the liver. Hepatic macrophages facilitate lymphoid organization and the maturation of B cells and T cells in response in the adult liver, and that lymphoid organization around hepatic macrophages is impaired in the young mice. So the implications from that is that age-dependent development of hepatic macrophages facilitates lymphoid organization in the liver and an effective immune response to HBV, and that there's, should be 2, I think that's one of the transfer issues to a PC. Age-dependent differences in hepatic macrophage expression of molecules in lymphoid organization and immune priming underlies the impaired immune response to HBV in mice. So we now have a list of molecules that we know are expressed in an age-dependent manner, minimally expressed in the young and expressed in the adult liver, particularly on macrophages and on antigen-presenting cells. And I'm just going to tell you about 2 of them, but I'm actually not going to show you the data because we don't have time. So the first one is CXCL13. It's a chemokine that's expressed by follicular stromal cells and some macrophages and conventional DCs. In the liver, it's expressed exclusively on the macrophages, and it's expressed on macrophages of adult mice and almost undetectable in the young mice. CXCR5 is its receptor. It's expressed on mature B cells and T follicular helper cells. CXCL13 directs B cell and T follicular helper cell homing to lymphoid follicles, and it's crucial in organizing cells within the follicle and follicular development and facilitating T and B cell interactions. Turns out it's very important in this liver organization. This is published work, but basically in the absence of CXCL13 or CXCR5, you get an immune response in adult mice that mimics viral persistence. So it's expressed in an age-dependent manner in mice, and in the absence of this interaction, you get the ineffective immune response that goes along with viral persistence. The other molecule that is expressed in an age-dependent manner is OX40 ligand. It's a member of the TNF superfamily. The ligand is expressed on professional APCs. Its cognate receptor is found on CD4, CD8s, and T follicular helper cells, and it's important in the division and survival of CD4 and CD8 T cells in long-term memory. And again, this is expressed in the antigen-presenting cells of adult liver and minimally in the young liver. And in the absence of OX40 or OX40 ligand, you absolutely phenocopy viral persistence, including the diminished IL-21. So our working model, based on this data, for persistence and clearance, is that in order to get effective viral clearance, you have to have this lymphoid organization that's anchored by CXCL13 positive macrophages and OX40 ligand expressing APCs. OX40 ligand is also on dendritic cells in the liver, which leads to priming of T follicular helper cells, production of IL-21, which leads to expansion and differentiation of the B cell responses and a broad, multispecific, long-lived T cell response, and the signature and lifelong viral clearance. In the young mice, you still get priming of a response, but because the macrophages don't express these molecules and some other molecules we've identified, the lymphoid organization doesn't occur. This whole lymphoid structure doesn't develop, and so the immune response is primed in the absence of T follicular helper cell activation, in the absence of IL-21, so you get an impaired expansion and differentiation of B cells, narrow and short-lived T cells, and the signature of viral persistence. So again, I just want to say that we validated these molecules in humans. CXCL13 is also expressed in an age-dependent manner in humans. There's increased plasma CXCL13 that's part of an effective immune response to HBV in adults acutely infected who clear. We don't see it in the flares or normals. And very interestingly, CXCL13 in chronically infected livers is at the same low level as infants. So it never goes up to the adult levels, really suggesting that this might be a therapeutic target. OX40 ligand is also expressed in an age-dependent manner in humans, just like mice. And patients with acute hepatitis B have significantly increased CD4 cells and CD4 cells that are OX40 positive compared to flares inactive and healthy patients. And so our conclusions are that immune priming to HBV occurs in the liver. Effective immune priming to HBV in the liver is impaired in young mice and humans. Lymphoid organization centered around CXCL13 positive macrophages and including OX40 ligand expression on hepatic APCs is crucial for immune priming and an effective immune response to HBV. And we've identified potential therapeutic targets that tilt the immune response in chronic infection towards clearance. So the last two very quick slides, we're working a lot on these as therapeutic targets, but what I want to show you is that young mice that are treated with the OX40 agonist develop hepatitis, have increased numbers of CD4 cells, and have increased numbers of T follicular helper cells. In these young mice that receive the agonist compared to the IgG control, 50% of them clear antigen lifelong. The clearance correlates with an increased diversity of the T cell response. And if we compare the mice that didn't clear, the young mice that got the agonist, the young mice, the 50% that cleared and the 50% that didn't, what you can see is those that cleared the ones that didn't clear still had the young response, so a response to the dominant epitope, but not the diverse response seen here. So the agonist by itself can actually lead to increased T cell diversity and antigen clearance. So our conclusions are that OX40 agonists can tilt the immune response in young mice towards HBV clearance. We're obviously working towards using mice that have seen antigen young and become adult and trying to get them to clear, because that's our target population, those that are chronically infected, the 400 million people. And I'll stop there and just thank all the people and institutions that helped us, particularly Jean Publicover, a very talented now associate scientist in the laboratory who helped develop the model and has been working on this a long time. Audra Johnson, Anuj Gagar, and Amanda Goodsell, my other collaborators at UCSF, CPMC, as well as I want to acknowledge the UCSF Liver Center, which has been instrumental in helping fund this work.
Within the last week, there were reports of successful immunization of infants born to HBV-positive moms with a conventional vaccine, suggesting that it's a matter of presentation then, I guess, of the surface antigen, or would you comment on how you think it fits with your model?
So it's been known that you can prevent infection in young infants by giving the vaccine. So the vaccine is standard of care as well as passive immunoglobulin. So, and the reason that that works is because when the antigen and when the vaccine is given, it's presented in the conventional lymphoid organs, the spleen and lymph node, which doesn't have this lag in proper problems with antigen presentation. So it's only when the antigen is first seen and presented in the liver that there's this age-dependent difference. And so if you can get the infant vaccinated, in fact, we vaccinate all our young children at birth against the vaccine, they can mount an effective immune response. It's true for our 3-week-old mice. If we vaccinate them, they make a perfectly good response. But in humans, if you're infected first and the antigen has been presented in the liver, that actually has global ramifications for the systemic immune response. You can vaccinate with the vaccine till the cows come home, and nothing will happen. So we know that we can prevent infection that way, but it's still a major problem in more rural areas of Southeast Asia where those protocols don't take place. Yes?
In animals that have species-specific hepatinoviruses, woodchucks, California ground squirrels, et cetera, how similar is their clinical and immunological response? And if you look to see whether these are true in the native models as well?
So the only native model we've looked in is humans. And in the woodchuck models, the presentation and age-dependent differences are similar, but it's very, very difficult to study the immunology and molecule expression in woodchucks. So you can look at gene expression, and we have some collaborators who work on woodchucks who are looking at some of the signature genes in that model, but the only native model that we've looked at is humans.
Hi, I'm just curious, a little bit naive to hepatitis B virus, but have you looked at other members of the L21 cytokine family, namely IL-7, IL-15, to see what the effect would be there, if it's relevant at all?
We've done panels looking for different cytokine expression, and there are likely other cytokines involved. We haven't found any as significantly different as IL-21 in this situation. I mean, there are some differences in gamma interferon, TNF production, but they're produced in the young. They're just produced at lower levels. But I certainly am not saying that IL-21 is the only pivotal cytokine in this process, but it's the one that likely contributes significantly to this difference in disease outcome in adult and young.
How specific is this to HBV, and is it also applying to other liver infections?
So that's a really good question. So this whole age-dependent development is age-dependent and not HBV-dependent. So we see you know, you don't have to be infected with the virus to have this difference, and we see it in wild-type mice. So the idea is that the mechanisms that we're identifying would be applicable to antigens that are first presented in the liver. So hepatotropic viruses, also things like malaria, you know, that kind of thing. But we think this is going to be universal to expression of pathogens that are first seen in the liver.
So, really nice talk. And
Thanks.
I wonder, have you ever tried putting the young T cells and B cells into an adult mouse, and how do they behave?
So they still get hepatitis, they still develop a diverse T cell response, but they don't, it's not as complete as putting adult cells in. There's some seroconversion and some not. So there are probably some defects in the age of the immune system, but not as profound as the age of the hepatic microenvironment in which the immune response is primed. But we don't know the details of that. But it's pretty close to an adult response, but there are some defects. It's not, you know, doesn't mirror the adult response.
Just a follow-up to that. In terms of the young mice versus the old mice, have you ever tried to sort of age the young cells by giving them, say, another virus, like LCMV or something like that? Specifically, do you know what it is about maturity that's driving?
So the cells that we believe that need to be aged, right, are the macrophages and antigen-presenting cells within the liver. So our experiments are about trying to drive those cells to express the molecules that we know to be important in this process. And so I just showed you the OX40 agonist stuff, but we're of course trying to just modulate the hepatic microenvironment to make it look more adult. And we've had quite a bit of success with that. Sorry.
From global health point of view, so because your model is kind of age dependent, and for the human beings, for example, in industrial countries, all of us receive the vaccine at our early age. But in developing countries, so currently we are promoting the vaccination of the adults. So I just want to see, can you make some comments for your model, you know, because in the developing countries, we give the vaccine to the adults. So their host defense to the vaccination is different than the children. So what kind of connection or the significance of your research and for that kind of point of view?
So the vaccine actually works well in young and in adult. So the vaccine works perfectly well in adult and perfectly well in young. So if I understand your question correctly, the significance in our work is, one, we're trying to understand the immune response that we're trying to achieve in those chronically infected. So we're trying to make it so that potentially we could have other ways of modulating the hepatic microenvironment that might not require vaccine. For example, changes in microbiome, et cetera, and to get young to clear initially. But really the other target audience that we're really thinking about here is we'd like to take our chronically infected adults who we know they were primed in the wrong way early, and we want to know how to switch them to make the immune response that leads to lifelong clearance. And I think that's the significance of the work. Thank you.